Thank you very much. I'll begin where Dr. Aaron ended, and thank Dr. Reese and the EMA personnel for organizing this workshop and for inviting us to attend. We're looking forward to a productive day. I'll begin with an overview, which will include the Orphan Drug Act in the United States. I'll talk about orphan drugs, rare diseases, and the Orphan Drug Designation Program, including requests, review of criteria, and benefits. The Orphan Drug Act was passed in 1983 to promote drug development in the United States. Prior to that, there had been about one drug passed for rare diseases per year, roughly, and Congress decided to try and provide incentives to increase that number. In January of last year, we celebrated the 30th year anniversary of the Orphan Drug Act, and we have at this point designated over 2,100 products, and over 460 of those have been approved. I'll start with some basic definitions. First, what is an orphan drug? In the United States, in the Code of Federal Regulations, and I won't do this all the time, but in CFR 316, it actually says, an orphan drug is a drug intended for use in a rare disease or condition. Designation is not mentioned. Note, being an orphan drug is not synonymous with having an orphan drug designation in the United States. What is a rare disease? Generally, a disease or condition that affects less than 200,000 people in the United States is that definition, and we'll talk more about that as I go on. So I'll begin by talking about the important actions that are pertinent to orphan drugs. The first being designation, and the second being the eventual new drug application for drugs and a biological licensing application approval for biologics. So what is orphan drug designation? In general, a drug or biologic may be designated by the Office of Orphan Products Development if it is to prevent, treat, or diagnose it as a disease or condition that occurs in less than 200,000 people in the U.S. The benefits of designation are financial incentives, essentially, including tax credits, where there's 50% of your clinical trial costs can be without tax. There's a waiver of marketing application user fees. We don't have a user fee for orphan drug designation, but for approvals we do, as you probably know. These are over $2 million for 2014, so it's a significant incentive. And we have 7-year marketing exclusivity. You're eligible for that if you have designation, if you're the first drug approved at that time for that indication. The other action is the NDA or BLA approval. The marketing approval is via the 505B and the Federal Food Drug and Cosmetic Act, or for a biologic, it's for approval under Section 351 of the Public Health Service Act. And in continuing to try and focus on the differences in these two actions, we'll talk about the organizations that make these decisions. In the FDA, the Office of the Commissioner is divided into a number of groups, one of which is the Office of Medical Products and Tobacco. That organization is divided into the Office of Special Medical Programs, the Center for Drug Evaluation and Research, and the Center for Biological Evaluation and Research, which are the two review groups that analyze the NDA and BLA applications. As you can see at the top, the Gold Star organization is the Office of Orphan Products Development. And as you can see in step 1, it makes the decisions regarding orphan designation. Later, when the NDA or BLA applications come in, the decisions are made by the Center for Drugs Evaluation and Research and the Center for Biologics Evaluation and Research. These are what we call the review divisions. And I'm going through this to point out that these are two different things. There's still people who think that orphan designation is tantamount to approval, or that it was likely to be approval. It's not the case. In part that is because the orphan designation decision is often made early in the developmental timeline. The designation request can be made during the preclinical development stage, prior to any clinical information, during the clinical development stage, certainly. In the United States, it must be made, the application must be made before submission of the NDA or the BLA. And also, it's interesting to note that no IND is required for orphan designation. All right, I'd like to talk a little bit about what we require or ask for when you make your application. First, as was stated earlier, we also use the common EMA FDA document, or you can submit it without that document. It's your choice. The first thing that we do ask for, that we require, is a simple statement of what the rare disease or condition is that you want to provide the therapy for. The second thing, we want you to identify the sponsor yourselves, contact information and the like, and the drug. The third thing is relatively important. This is where you provide your background about the disease or condition. We want to know how the drug is proposed to be used, and we would like to know the reasons why you think such therapy is needed. The fourth request is for a detailed description of the drug. We don't, or we frown upon, and we really don't accept an internal number or code as your indicator. We need to know what you're actually talking about in terms of what your drug is. And so we need a little bit of a detailed description. And then we also want to know what the scientific rationale is for the use of the drug or biologic. Our fifth request has to do with the same drug termination. We'll talk, Dr. Milter will talk later today more about the same drugs. We asked that if you think the drug is the same drug as an already approved drug, with or without orphan exclusivity, we ask that you tell us why your drug should be clinically superior. The question may come up, that sometimes what we think is the same drug and what you think the same drug can differ, and that becomes complex. But basically, the whole point is once the FDA has decided that the drug is the same drug, and it's already approved, then you have to provide information about the clinical superiority in order to get designation. Number 6 is asking whether the disease or condition that you're considering is a subset of a common disease, and what the basis is that you think it is. In a final rule, which I'll talk a little bit at the end of the study, which was published last year, we emphasize that an orphan subset needs to be based on a characteristic of the drug which limits its use to the subset in question, and by that characteristic wouldn't be used in the other part of the common disease. And again, this is in the final rule, which is published on our website, www.fda.gov/orphan. And again, I'll talk a little bit more about that later, but it fits into this number 6 item. Number 7 is we asked for a summary of the regulatory status and the marketing history of the drug or biologic. And 8 is your documentation, references, calculations about the prevalence and why you think it's less than 200,000. Or the other possibility is where you would show that there's no reasonable expectation that the costs of research and development of the drug can be recovered by sales. This is an extremely rare basis to make a designation. It's only happened 2 or 3 times in the history. I don't have that number exactly, but it's basically 2 or 3 times of the Office of Orphan Products Development. Okay. So having submitted your request, what is the procedures that we do in terms of making a review of the designation request? There are three main questions. What is the disease or condition? Is the disease rare, i.e. what's the prevalence? And is there sufficient scientific rationale that demonstrates promise that the drug or biologic will treat, diagnose, or prevent the disease or condition? Again, we say promise at this point because the designation is frequently very early in development, and we don't have endpoint clinical trial information to deal with. We're getting a suggestion that something is going on, and that it's a good idea to proceed with the developmental investigations. So the first question is, what is the disease or condition? Basically, we have to determine the disease or what we think the disease or condition that's you're treating. In other words, if you come in and say, we'd like to focus on some aspect of such and so disease, and therefore we're treating such and so disease. The aspect that you're treating might be pain. If that pain and the mechanism for treating it via your drug could also be used for other pain, similar pain in other situations, we would say you're treating pain, not the disease. So we would determine that what you're actually treating is the pain and not that particular potentially orphan disease. In terms of looking at the evaluation of diseases can be challenging and can change over time. For instance, scleroderma used to be considered one disease by FDA. And occasionally people would come in for either localized scleroderma or systemic sclerosis as a subset to be considered. At this point, after consultations with the review divisions, we now look at localized scleroderma and systemic sclerosis as potentially, or I'm sorry, not as different diseases to be evaluated. Oh, I'm sorry, I didn't change the slide there. So again, scleroderma used to be the individual disease, and the two others are listed. I apologize. Continuing with the question about is the disease rare, this is the various ways that we look at whether the drug is for treatment, for diagnostic claims, or prevention claims. And it does differ slightly. For treatments, it's the overall prevalence of the people with the disease in the US at the time of submission. And it needs to be less than 200,000. For example, United States, one disease that would qualify under that circumstance, an example would be sickle cell disease. Now, of course, sickle cell disease is not rare in other parts of the world, but it is in the United States. An exception to that is for acute illnesses, which we consider to have a duration of less than 1 year, in which we use incidence. So if the diseases occurred, we would count the number of diseases that occur per year. An example would be, say, necrotizing soft tissue infections. For diagnostic claims, we consider all those people who would be subjected to diagnosis per year, of course, in the US. And an example would be if the drug was supposed to be a confirmatory diagnostic for anthrax, we're clearly not going to give everybody in the country a diagnostic for anthrax, but there'd be we would have to have some basis for using the diagnostic, and that'd be far less than obviously the 200,000 likely. For prevention claims, we will look at everyone who is at risk of the disease per year. So, for the example of prevention of corneal transplant rejection, the people would be at risk for the rejection are obviously people who have had corneal transplants. So that would be the basis for the prevalence decision. In terms of the prevalence estimate, we ask that you give us a specific number when you make your request. Sometimes, not infrequently, sponsors will say, well, here's our information, we think it's less than 200,000. We pretty much demand, sometimes not, but pretty much demand that you give us a number that we can have for the record. Examples of sources to use to calculate prevalence include published literature, for example, of clinical trials, say. Certainly, that's not the case necessarily when you make a very early request, but it's something you can do. Registries, in terms of patient registries, the SEER database, which is surveillance, epidemiology, and end results. It's put out by the NIH for oncology indications. And if those three, if you can't come up with anything from those three, we do allow 3 independent expert opinions, but it's sort of the last option that we will accept. In terms of the range, sometimes people provide ranges of prevalence estimates, so they get several estimates from several sources. And sometimes they want to use the average or something along those lines. For example, in myasthenia gravis, we have prevalence would be anywhere from 43,500 to 63,500. The FDA's standing consideration is that we always choose the largest number. So we don't do averages. We always choose the largest number, even if we have to find it. Now I'm going to talk a little bit about orphan subsets relative to rare diseases. And of course, if the disease is common, has more than 200,000 persons, we can grant designation as an orphan subset. But the basis, as I alluded to earlier, is that a subset of all persons with a disease condition would only be expected to benefit from the drug. So if there's a small group that will benefit from the drug and the rest of the common disease won't, that likely will qualify, of course, depending on the specifics of the case. An example would be, for a common disease, non-small cell lung cancer, which is not an orphan. The orphan subset could be non-small cell lung cancer associated with an epidermal growth factor receptor mutation. Now, that type of cancer with that mutation, the drug or biologic would have to interact with that receptor in some manner that it wouldn't interact with other non-small cell lung cancers. And so the qualification would be that the characteristic of the drug or biologic limits its use to the people with a mutation, and that would not be effective in the people with non-small cell lung cancer in general. A little more about orphan subsets. We don't appreciate or like what we call salami slicing. The idea that if you represent a salami, a large common disease, and you would slice it up into subgroups, if you will, based on some criteria or other. An example of what we wouldn't accept would be if someone comes in and said, we'd like to use our drug for breast cancer only for patients refractory to first-line treatment. We would say no to that unless there was some property of the drug that limited it to the people that you wanted to use it in, and it would not be useful, again, in the larger population. Okay? Another example would be if you came in and said, we'd like to use a subset based on the clinical trial inclusion criteria. That, again, unless something about the drug limited to other people with that same common disease, we wouldn't accept that. Some subsets examples that we might accept, again, a case-by-case basis, would be if there was a drug or monoclonal antibody against a surface antigen particular to a subset or a population with breast disease that only had that surface antigen. And your drug would interact with that particular surface antigen, i.e. it would be limited to the use in that population, and it would not interact with other types of breast cancer. That would be a basis for potentially making an orphan subset. Another example would be if there was a genetic mutation that the drug interacted with, like say making a subset of, say, colon cancer, and again, that drug would not work in other types of colon cancer without that particular genetic mutation. Okay, so then we turn to the third main question, is the scientific rationale sufficient? Again, what's required is evidence that the drug holds promise for being effective in treating, preventing, or diagnosing disease. And it can include information from clinical data, animal models, or less frequently, in vitro data, as I'll allude to momentarily. I think here it's good to use, to include a sentence that is frequently used in our deficiency letters when the deficiency has to do with a scientific rationale deficiency. And that is, the scientific rationale is best supported by clinical data. So, well, that's the human data is the best information. If you have clinical trial data, that's great. Frequently we don't because, again, designation is early in the developmental timeline. But if you're at the stage, say, in an oncology situation, sometimes they'll do uncontrolled trials with patients who have tried everything else available, and they basically give them the new drug just to see what might happen. If even in a handful of patients we find that there's some partial responses or complete responses, that has been in the past considered as adequate for clinical information. And then the sentence goes on. However, in the absence of human data, the application for orphan drug designation may be satisfactorily supported with a relevant animal model for the human disease. That really means an animal model of the specific disease that you're looking at. It doesn't mean a model that's similar or that a model that probably works with the same mechanism that your drug is supposed to affect. It really means an animal model an accepted animal model of the disease that you're talking about. So that's something to keep in mind. In terms of scientific rationale, a recent analysis showed that for designations in a given year, 2/3 of the time the designation decision was made on clinical experience. A full 1/3 of the time it was made on animal study data, and at 2% of the time, in very unusual cases, we worked with in vitro study data when there was no accepted animal model at all for the particular disease. So what can you expect after you've sent in your request? The typical review cycle is 90 days, although we don't have an absolute limit. We certainly try to meet the 90-day, our requirement of 90 days. At the end of the cycle, you'll receive either a designation letter or a deficiency letter. And once designated, you as a sponsor are required to submit annual reports until the drug is approved. I'm going to jump ahead for a moment here to the end of the approval period. So, assuming you've had designation and you now had an approval, it's not uncommon that the designation terminology is not identical to the labeled indication passed by the review divisions. Remember, the review divisions are not the Office of Orphan Products Development, and they may approve you for something somewhat different than what we wrote the designation for. And that has to be evaluated at the time in terms of whether exclusivity is available based on our designation wording and that wording. But in the case of looking at when a labeled indication is narrower, for example, there's a recent example. Bosutinib was designated for the treatment of chronic myelogenous leukemia. That was our wording, treatment of chronic myelogenous leukemia. When it was approved, bosutinib was approved for treatment of Philadelphia chromosome-positive chronic myelogenous leukemia with resistance or intolerance to prior therapy. So it was an indication that was less than the broader designation indication. But we considered, as you can see from the diagram, that the approved labeled indication was covered by the orphan designation. So even though the wording was not exact, that understanding was recognized. So how have we done? This is a a chart with the timeline, if you will, from 1983 through 2013, showing drug designations and drug approvals. Last year in 2013, the designations was, the number was 258. The requests was approximately 350, thereabouts, but the designations we're talking about 258. The approvals, as you can see, the black bars, were 33. Keep in mind that the approvals didn't come in 2013. The approvals, there's an obvious lag time of a few years or maybe longer, certainly longer. So really what's going on in 2013 is probably, well, there's no question, is from the years before that. So we would think that in the next few years, with the big increase in designations, that the approvals should be increasing over time. I also can say that in terms of designation requests, for 2014, we're already beating the pace for 2013 in terms of requests, which of course is not on this slide. Another analysis is this pie chart looking at orphan drug designations in 2013. Similar to what was said by Dr. Aram, the oncology designations were the highest with 38%. Neurology was 12%. Hematology, metabolism, infectious disease, gastroenterology, and pulmonary between 7 and 4%. And the other 22% who had a lower than 4% designation number included a full 1/5 of the designations. The final thing I want to talk about briefly, is the final rule. Our final rule is our term for the final publication of basically rule changes, regulation changes that we do. The most recent was in August 12th, 2013. The previous one was in 1992. So it's not a common phenomenon. And in this final rule, it was a group of amendments intended to clarify regulatory language and areas of minor improvement regarding orphan drug designation and exclusivity in terms of the written regulations. I'm going to give you just two quick examples. If you have a situation where a sponsor has a drug with exclusive approval for only one indication within that designated disease, okay? So there's a designated disease, the approval is for one indication within that designated disease. And then for some reason or another, they come along and find with more information that there can be more indications within the disease that could be approved. We will recognize the orphan drug exclusive approval of those extra indications. Okay, so the problem would have been we approved an indication and then another one came along by the same company. We would approve the extra indication if it fell under the designation. It may not be easy to grasp all these things right as I'm saying about them. But, the fact is, the final rule is on the website, and you can read the specific details and the preamble so you can understand the thinking that led to these changes. One other fine-tuning was a clarification that if you've already, we already know that a sponsor can't ask for an orphan designation after marketing application is in. But it was clarified that if the marketing application of one sponsor is in-house, it doesn't block another sponsor from getting designation for the same drug for the same indication based on the marketing application that's already in-house for the first sponsor. The idea was we didn't want the first sponsor to have marketing application and be developing the drug or biologic. But if somebody else came along, well, probably a drug, but somebody else with the drug came along and wanted to develop the drug for the same indication, and we didn't want to limit their possibility perhaps to develop it more quickly, by the fact that the first sponsor's marketing application was in-house. So they can get designation, and then the marketing application exclusivity would be available, they would be eligible for it, depending on who got the first approval. Again, these things are all better described than I just did in on the website. Oh, I'm sorry, I missed getting carried away with not doing the slides here. So the last two slides about the final rule had to do with those two situations. So we have a website, www.fda.gov/orphan, and you can email us at orphan@fda.hhs.gov or 301-796-8660 if you have further questions, and of course we'll be around all day. Thank you.